Assalamu alaikum wa rahmatullahi wa barakatuhu, and good day to each and everyone. I am Arifa C. Akub from Bio 107 Section CDE, and today I will be presenting to you a study entitled Hepatic de novo lipogenesis is suppressed and fat oxidation is increased by omega-3 fatty acids at the expense of glucose metabolism. So this is a work of J. Green and others. So this presentation contains first, the background information of the said study, second, the methodology, third, the results and discussions, and lastly, the conclusions. Before I begin to discuss the study, let us first define some important terms that you need to know for you to be able to understand the study. So first, the hepatic de novo lipogenesis. So this is the biochemical process of synthesizing fatty acid from acetyl-CoA subunits that are produced from a number of different pathways within the cell, most commonly carbohydrate catabolism. Next, non-alcoholic fatty liver disease. It is an umbrella term for a range of liver conditions affecting people who drink little to no alcohol. As the name implies, the main characteristic of non-alcoholic fatty liver disease is too much fat stored in liver cells. Next is the triacylglycerol. It is an ester derived from glycerol and 3 fatty acids. Fat oxidation, it refers to the process of breaking down fatty acids. And lastly, omega-3 fatty acid. It is a fatty acid with multiple double bonds. Non-alcoholic fatty liver disease, defined as excess intrahepatocellular triacylglycerol, is an accumulation due to non-alcoholic causes. It is a complication in individuals who are obese and or have type 2 diabetes. Increased hepatic de novo lipogenesis is often suggested to be an underlying cause in the development of the NAFLD and or insulin resistance. Observational studies have reported fasting hepatic DNL to be higher in individuals with NAFLD compared with those without. It has been suggested that changes in plasma TAG concentration are proportional to the amounts of hepatic DNL. It is suggested that omega-3 fatty acid lower hepatic DNL. So that is the focus of this study, to experiment about the omega-3 fatty acid effects on the hepatic DNL. The researchers investigated the effects of omega-3 fatty acid supplementation on hepatic DNL and fatty acid oxidation using a combination of human in vivo and in vitro studies. So for the methodology, there were 38 healthy men that were the subject of this study that were randomized to take either an omega-3 supplement, that is the focus of this study, or that is what we are trying to find out about its effect, or a placebo. A placebo is an inactive substance that is used to suggest whether changes in the test group result from the treatment or it occurred by chance. And also, fasting measurements were made at baseline and after 8 weeks. The metabolic effects of omega-3 fatty acids on intrahepatocellular triacylglycerol content, hepatic DNL, and fatty acid oxidation were investigated using metabolic substrates labeled with stable isotope tracers. In vitro studies using a human liver cell line was undertaken to gain insight into the intrahepatocellular effects of the omega-3 fatty acids. So this is the results and discussion of the study. This table shows the characteristic of the study participants at the baseline and after 8 weeks. So there were 38 subjects. So 19 is from placebo and 19 take the omega-3 fatty acid. So for the age, weight, BMI, and the weight circumference between or within the groups at the baseline or after 8 weeks, there was no difference, or there is no significant changes. There was also no difference between groups in body composition, which was assessed at the baseline visit with total fat, lean, and visceral masses. So as you can see, the only thing that significantly changed is the tag in the fasting plasma biochemical parameter, the tag or the triacylglycerol. So as you can see, there is 2 asterisks, which means that there is a significant change in this, meaning that it decreases. So another thing that decreases is this one, the alt. The alt also has 2 asterisks here, which means that it significantly decreased after 8 weeks. And another thing that we can observe is the glucose. The fasting glucose has 1 asterisk, which means that it significantly increased. So now for the table 2, it shows the intrahepatocellular triacylglycerol content and fasting and postprandial biochemical characteristic of participant taking omega-3 fatty acid. So there are a total of 19 who took the omega-3 fatty acid. As you can see, the intrahepatocellular triacylglycerol, which is the focus of this study, to lower this is the focus of this study, it has significant changes. So by taking omega-3 fatty acid, this triacylglycerol, which is a fatty acid, it actually decreases, which means that the omega-3 fatty acid is effective. So below here we can see the fasting plasma biochemical parameter and then the postprandial plasma biochemical parameter, which is the in vitro study. So for the VLDL, these are VLDL, they are very low density lipoprotein, a combination of lipids and protein. These are actually made in the liver, and the target of this is the plasma and also in the liver. And the major composition of this, the major lipid composition of this VLDL is actually the cholesterol. As you can see, there is a significant change in the triacylglycerol, and also for the tag and VLDL ApoB, which means that this lipid decrease by taking omega-3 fatty acid. And then this one is, compared to the fasting, this is the postprandial. As you can see, there is also a significant change only in the tag. In the chylomicron, which is also a lipid which has a major composition of triacylglycerol, there is no significant change. These also, the NEFA and the 3-OHB. The NEFA is the non-esterified fatty acid, so there is no significant change. And then lastly is the postprandial C-label plasma biochemical parameter. So this C-label here means that this one is the in vitro studies. So I have mentioned earlier that this study uses in vivo and the in vitro. So this one is the in vitro studies that uses a human cell liver that is undertaken to gain insight into the intrahepatocellular effects of the omega-3 fatty acid. As you can also observe, the triacylglycerol also has significant change, which means that really the omega-3 fatty acid has an effect on the IH tag percentage in the body. So So this is now the summary of the results. So fasting plasma TAG concentration significantly decreased in the omega-3 group and remain unchanged in the placebo group. 8 weeks of omega-3 supplementation significantly decreased age TAG, fasting, and postprandial hepatic DNL, while significantly increasing dietary fatty acid oxidation and fasting and postprandial plasma glucose concentration. In vitro studies supported the in vivo findings of omega-3 fatty acids, decreasing intracellular triacylglycerol through a shift in cellular metabolism away from fatty acid esterification toward oxidation. For the conclusion of the study, so omega-3 supplementation had a potent effect on decreasing hepatic DNL and increasing fatty acid oxidation and plasma glucose concentrations. Attenuation of the hepatic DNL may be considered advantageous. However, consideration is required as to what the potential excess of non-lipid substrate, for example, the glucose, will have on the intrahepatic and extrahepatic metabolic pathways. That's all. Thank you for listening.